Welcome to plus 2 first year science. I am R.K. Pradhan, Department of Botany, Higher Secondary School, Barampur. Last class we discussed about different type of proteins and their structure and function, and also we discussed about the lipids. Lipids consist of two parts, that is the fatty acid and glycerol. We have already discussed about fatty acid and glycerol, types of fatty acid, that is saturated and unsaturated fatty acid. We have already discussed about all types of fatty acid and glycerol, but lipids are classified into three types, that is simple fats or simple fat, compound lipid, and a derived lipid. Today I will discuss about the classification of lipid. Then come to the smart board. First we have to know the classification of lipid. Lipid generally classified into three groups, that is simple lipid, compound lipid, and derived lipid. Simple lipids are includes mainly two things. One is fats and wax. Fats and wax or oils are the simple lipids. They specially made of fatty acid and glycerol. First we have to discuss about the fats. Fats are the simple lipid, that is if glycerols having three fatty acids, then that will produce 3 molecule of ATP, 3 molecule of water. But actually then generally fats are solid at room temperature, means at 20 degree Celsius temperature fats are generally solid. Why? Because they have more saturated fatty acid. If more saturated fatty acids are present, they have higher melting point, means they become solid at room temperature. Generally fats are solid at room temperature because they have more saturated fatty acid. Saturated fatty acids having single bond, single covalent bond, that means they are fully saturated with a hydrogen atom. So they are definitely solid at room temperature. Then another thing is short chain of fats and unsaturated fatty acid having low melting point, means those fats having unsaturated fatty acids and short chain, they have low melting point, means they can easily melted at low temperature. So definitely they have low melting point. Then another point is rancidity. You know, this is the property of fats, rancidity. Rancidity is the unpleasant odor and taste of the fat due to long time preservation, due to the microbial degradation activity, and also if they will expose to air, moisture, and a different contamination of a microorganism, that causes unpleasant odor, means that fats becomes having unpleasant odor and having bad taste. So this property of fats, that is called as rancidity. Generally animal fats having higher rancidity, means they have more, they can expose to, if they can expose to air and moisture, they have easily unpleasant odor or and bad taste and smell, so that can not be used by the human being. This is only due to the microbial activity, degradation activity. So generally a plant fat having low rancidity property. Why? Because of presence of vitamin E. You know, tocopherol is a compound, that is a vitamin E. Presence of tocopherol in the plant fat, that reduces the rancidity of the plant fat. But animal fats or butters having more rancidity, they cannot be preserved for long time, so that can be degraded by the microbial activity as well as a exposure of air and moisture. So they have more rancidity property as compared to plant fat. Then comes to depending upon the number of fatty acid, fats are generally three types. First one is monoglyceride, second is diglyceride, third is triglyceride. Monoglyceride means what? Mono means single. If single fatty acid present in the glycerol, that is called as monoglyceride. If one fatty acid plus glycerol, that form the monoglyceride. Similarly, diglyceride, two fatty acid, when two fatty acid combine with glycerol, that results in the formation of diglyceride. Same when three fatty acid combine with glycerol, that results in the triglyceride. Then you see the see the picture of the board. Here this is glycerol, means here 3 OH group is present. This is first OH, this is second OH, this is third OH, hydroxyl group. But here 3 fatty acids are also present. 3 fatty acids are present. When 3 fatty acids combines with glycerol, that results in the formation of triglyceride. Here you see the 3 ester bond is formed. One fatty acid combines with a hydroxyl group of glycerol, that form one ester bond. One ester bond. Then another fatty acid combines with OH group, that also from second ester bond. Then another fatty acid combines with OH group, third OH group, that results in the formation of third ester bond. In this way, 3 ester bond formed, resulting formation of 3 water molecule. Here one water molecule release, here another one water molecule release, here another water molecule release. When 3 fatty acid combines with a glycerol molecule, that results in the liberation or release of 3 molecule of water. This is all about your fats. Then comes to second type, that is wax. Here ester of long chain of fatty acid with monohydroxylic alcohol. Here wax or oils are the simple lipid, that consists of ester of long chain of fatty acid with monohydroxylic alcohol, means single OH group present in the glycerol, that combines with a long chain of fatty acid, that results in the formation of wax. Fatty acid and alcohol, that results in the formation of lipids. Generally we have to see the when fatty acid combines with alcohol in waxes, that varies from 24 to 26 carbon. We know fats or waxes are waterproof coating that form on the outer surface of the plant body as well as animal body. Generally, wax are also solid at room temperature. Different type of waxes are produced in different animal body. Example, first one is bee wax. You know, bee wax means these are the waxes produced from the abdominal cavity, abdominal gland of the honeybee, male worker honeybee, that is required for formation of honeycomb. Bee wax is a wax or a simple lipid substance generally secreted from the abdominal gland of the male worker of honeybee, that is required for construction of a honeycomb. Similarly, another is cerumen. Cerumen means that is ear wax. Wax is a fats which are also secreted from the external auditory gland of the ear, that is deposited in the form of a waxy substance, that is called as cerumen, that is specially found in animal ears as well as human being. Then another thing is sebum. You know sebum is a waxy substance secreted from the sebaceous gland of the skin. In our skin there are different sebaceous glands are present. They secretes waxy fatty substance that is called as sebum. Then another thing is wool wax or linolein. Wool wax means they are specially secreted from the cutaneous glands of the insect. Surface of the skin, surface of the body surface, they are very much similar with the sebum. Wool wax or linolein, that is very much similar with the sebum, that is secreted from the sebaceous gland. Then another thing is spermaceti. Spermaceti means it is also a wax that is secreted from the head of the sperm of blue whale. That is a very hard substance secreted from the head of the sperm of whale, blue whale. That is called as spermaceti. Then another is a paraffin wax. Paraffin wax is a fatty substance or waxy substance that is also produced from the petroleum product, and candle is generally made from the paraffin wax. These are the different types of fatty substance or fats or simple lipids obtained from different types of animals, insect as well as human being also. Then comes to next. Then these are discussed about under simple lipid. Then second type is compound lipid. What is compound lipid? We have already discussed when fatty acid combines with alcohol or glycerol and some other additional group that form the compound lipid. Here fatty acid plus alcohol combines with additional group, that may be phosphoric acid, that may be protein, that may be any non-protein compounds which may be attached with the alcohol group and fatty acid, that resulting in the formation of compound lipid. Generally compound lipids are three types: glycolipid, phospholipid, lipoprotein. First one is glycolipid. Glyco means what? Glyco means glucose, lipid means lipid. When glucose combines with lipid, that results in the formation of glycolipid. Here fatty acid combines with sphingosine. What do you mean by sphingosine? That is amino alcohol. Amino alcohol is called as sphingosine and combines with oligosaccharide. Fatty acid, sphingosine and oligosaccharide, they constitute to form the glycolipid. It is a component of antigen A and B. Everybody knows antigen A and B which present on the surface of RBC. Human RBC contains antigen A and B, and so they create, and that they also react with antibody A and B. Antigen antibody reaction take place in our body. This antigen A and B is formed by the glycolipid. Similarly, gangliosides possess sugar residue like glucose, galactose, sialic acid combines with fatty acid and sphingosine. That also results in the formation of gangliosides. See, these gangliosides are found in the myelin sheath of the nerve fiber. We know myelin sheath is the covering sheath of nerve fiber present in the animal body. So, where myelin sheath is composed of gangliosides. Gangliosides composed of sugar, fatty acid and sphingosine. Sphingosine means amino alcohol. Then phospholipid. Second type is phospholipid, lipid with phosphate group. Lipids means fatty acid, glycerol and phosphoric acid. When they together constitute to form the phospholipid. Together constitute form the phospholipid, that is here lipid combines with the phosphate group or phosphoric acid, that results in the formation of phospholipid. Here third fatty acid is replaced by phosphate group and nitrogenous bases. First and second fatty acid remain present in the glycerol or remain attached with the glycerol, but third fatty acid replaced by the phosphoric acid or phosphate group and other nitrogenous bases, that results in the formation of phospholipid. Then choline is a nitrogenous basis which present in the lecithin. Similarly, ethanolamine present in the cephalin, and these phospholipids are amphipathic layer in cell membrane. That means the phospholipid bilayer that results in the formation of a phospholipid bilayer. You know, this is the hydrophospholipid molecule. Similarly, we can draw the another phospholipid layer, that is here this is hydrophilic head, this is hydrophobic tail. One phospholipid consists of two parts. One is hydrophilic head which are polar nature. Polar nature means they are soluble in water, but hydrophobic tail is non-polar nature, means they are insoluble in water. But these two layer phospholipid bilayer form the phospholipid bilayer in the cell membrane. I have already discussed about the structure and function of cell membrane. Here phospholipid bilayer is the important constituent of the cell membrane. Phospholipid means fatty acid plus glycerol plus phosphoric acid and nitrogenous bases like choline, ethanolamine, serine. These are the different type of nitrogenous bases which remain attached with the phosphate group. Here choline is a nitrogenous basis present in the lecithin. Ethanolamine is present in the nitrogen base of cephalin. So example, lecithin or phosphatidyl choline. Here lecithin is also called as phosphatidyl choline, means phosphoric acid plus fatty acid plus alcohol plus choline. Choline is a nitrogenous basis which remain attached with the phosphoric acid, that form the lecithin. This lecithin present in the cell membrane, egg yolk and oil seeds. You remember, lecithin present in cell membrane, egg yolk and oil seeds. Similarly, cephalin is also called as phosphatidyl ethanolamine. Phosphatidyl means fatty acid plus glycerol plus phosphoric acid, nitrogenous base, that is ethanolamine. These four components constitute the cephalin or phosphatidyl ethanolamine, which are present in nerve tissue and blood platelets. Similarly, another things, that is dipalmitoyl lecithin, that function as a surfactant in alveoli, means that protect the alveoli cells of the lungs. Dipalmitoyl lecithin, which is a phospholipid present on the alveoli, that prevent or protect from the different water condition of the lungs. So here this is the structure of the lecithin. Here this is CH2OR1. Here one fatty acid combines with OH. and second fatty acid combines with the OH, but third fatty acid is replaced by phosphoric acid. You know, this is a triglyceride. Here third fatty acid is replaced by phosphoric acid, and they are, that is combines with a lecithin. Lecithin is a nitrogenous basis which remain attached with the phosphoric acid, that results in the formation of dipalmitoyl lecithin, or that is also called as phosphatidyl choline. So this is the entire structure of the phospholipid. Mainly phospholipid is the basic component of the cell membrane. They are also present in the oil seed, blood platelets, and membrane of the nerve tissue, and also present around the alveoli, lung cell. So these are the very, very important component of the cell membrane. Then comes to lipoprotein. Third type of lipoprotein. Lipoprotein is a generally there are two components, that is lipid and protein. When lipid and proteins are combined with each other, that form lipoprotein. This is the third type of compound lipid, but they are mostly available in blood, milk, egg, yolk. But VLDL, LDL. VLDL means very low density lipoprotein, VLDL, and a low density lipoprotein LDL, and high density lipoprotein HDL. But those lipoproteins having high density, they are good fat, they are good lipid, means they have no harmful effect in our body. Those having higher density, they have no harmful effect in our body. Especially HDL is very, very good. They are the very good fat or lipid of our body. They have no harmful effect in our body. But these are all about your compound lipid. Then comes to derived lipid. Derived lipid means those lipids which are obtained or derived from simple and compound lipid on hydrolysis. Those lipids which are obtained from simple and compound lipids on hydrolysis, that is called as derived lipid. Then they don't contain any fatty acid but show fat properties. Generally there are two types, one is steroids, another is a chromolipid or terpenoids. Derived lipids are two types, one is steroids, second is chromolipid or terpenes. Actually steroids are they are the cyclopentano per hydrophenanthrin structure. They have 17 carbon atom. Specially they are called as, steroids are called as cyclopentano per hydrophenanthrin structure. But they are a 19 membered cyclic ring structure, nuclear structure, but they are of different types. First one is a cholesterol, ergosterol, stigmosterol. Sterols are of three types. One is cholesterol, second is stigmosterol, and but first cholesterol whose chemical formula is carbon 27, hydrogen 45, and OH. Here OH group is present, so the steroid compound is called as sterol. If OH or alcohol group is present, the steroid compound can be considered as a sterol. Al means alcohol group is present. Cholesterol compound or steroids are specially found in animal cell membrane They are specially also they are present in our adipose tissue in the form of globule. They function as a thermo insulator, means they they reserve our the thermo-insulating substance. Cholesterol act as a thermo-insulating substance in our body. So that that is also very much essential for our body, but whose concentration is, if its concentration is more in the blood, that will be very they have a highly harmful effect to our body. Then second type is ergosterol. Ergosterol is whose chemical formula is carbon 28, hydrogen 48 and OH. Ergosterol is specially found in fungi and yeast. Fungi and yeast, where ergosterols are present, they are the steroid compounds. They are found in fungi and yeast. And another is stigmosterol. Stigmosterols and also beta-sterol. Another thing is beta-sterol, which are found in plants. But this is the structure of cholesterol or steroid compound. It has a four ring structure. But that is named as A, B, C, D ring. But A, B, C rings are hexane ring. That is called as hexane ring or hexagonal structure. And a fourth ring, that is D ring, is considered as is a pentagonal ring, that is called as cyclopentane ring. That is hexane ring, that is cyclopentane ring. Four rings are connected with each other. They contain 17 carbon atom, but they are 19 membered ring structure. They contain different carbon atom. So whose number of carbon atom is 17, but 19 membrane ring structure. These are different types. This is the structure of sterol, and different type of sterols are found in different organisms. Cholesterol found in animals, ergosterol found in fungi and yeast, stigmosterol found in and beta-sterol found in plants. Then comes to bile salt. We know bile salt which are present in our gallbladder, that is sodium tercocholate and sodium glycocolate. They are also steroid compounds. They help in emulsification of fats. Bile salt help in emulsification of fat, means they help in breakdown of large fat globule into small fat globule. There are two types of bile salt, that is sodium tercholate and sodium glycolate. They help in emulsification of fats, means they help in breaking the large fat globule into small fat globule. Then another is sex hormone. Different type of sex hormones are there: progesterone, androgen, testosterone. They are the sex hormone which are the steroid compound. Especially cholesterol are the precursor of the sex hormone. Means these sex hormones are produced from the cholesterol. Cholesterols are the precursor for sex hormone like progesterone, androgen and testosterone. Then another is adrenohormone. Adrenohormone means they are secreted from the adrenal gland. Adenocortisone, adenomineral cortisone, they are the different type of hormone secreted from the adrenal gland, but specially they are steroid hormone, and cholesterol are the precursor of such type of hormone. Then another anabolic steroid. We know anabolic steroids are the compound which help in maintaining the growth and development of the bodies. Most especially athletes are using such type of steroid for making their body for building their body. So that have also harmful effect. If we will continuously use this steroid, that will have many disadvantage, the harmful effect to our body. So these are the different type of steroids are found in different organisms like plants, animals, and also they are found in different glands. Then second type is terpenes. We have already said derived lipids are two types, one is steroid, second is terpenes. Terpenes are the contains isoprenes. Isoprenes means it is a compound whose molecular formula is a carbon 5 and hydrogen 6. This is a hydrocarbon compound, carbon 5 hydrogen 6, C5H6, that is called as isoprene. These isoprenes are connected with each other that form the terpene or terpenoids. They are specially the derived lipid. Then carotenoids, we have already know what is carotenoids. Carotenoids are the plant pigments which are abundant in different green plant like carotene as well as xanthophyll. Carotene are the orange color pigment, xanthophyll are the yellow color pigment which are found in different algae. Carotene, beta carotene, alpha carotene, these are the derived lipid. Carotenoids pigments are the derived lipid. Specially they are found in these terpenes are specially found in essential oil, menthol, carotene, vitamin A, K, and natural rubber. These are the compound which are found in different substance of the plants. So terpenes and steroids are two derived lipid which are available in plants as well as animals. These are all, this is all about your lipid portion, simple lipid, compound lipid, and derived lipid. Then comes to today I will again start another chapter, that is enzyme. Enzyme everybody knows enzyme. Enzyme has a very, very important role in our body. Enzyme plays a key role in our body. Means they regulate the biochemical reaction of all living organisms. Without enzyme, nothing cannot be possible in our body. Enzyme plays vital key role in our body. Enzyme, especially enzyme, stimulate or accelerate the biochemical reaction of our living system, of our cell. Means those reaction or chemical reaction which take place in our living cell, that is called as biochemical reaction. That is also called as metabolism. Metabolism is of two types, that is anabolism and catabolism. Constructive process of the cell, constructive biochemical reaction of our cell, that is called as a anabolism. Destructive biochemical reaction of our cell, that is called as catabolism. Here anabolism and catabolism together constitute the metabolism of our cell. So enzymes are also called as metabolic regulator because they regulate the metabolism process of our cell. Different type of reaction which taking place inside our cell, different type of biochemical reaction which are taking place in our cell, they are regulated or controlled by the enzymes. So enzymes are also called as metabolic regulator. But actually enzyme means N plus zyme. Here enzyme means N plus zyme. N plus means N means in, zyme means yeast. Enzyme means in yeast. Means those substance present in yeast, that is called as enzyme. Enzyme is an organic compound which present inside the yeast cell, that is called as enzyme. But actually enzymes are biocatalyst. Bio means living, catalyst means catalyze the reaction, means they accelerate the reaction. Those substance or organic compound catalyze the biological reaction or biochemical reaction of a living cell, that is called as biocatalyst. Enzymes are also called as biocatalyst. Similarly, they are also called as metabolic regulator. In our cell, different type of reaction, chemical reaction take place like glycolytic reaction, Krebs cycle. They are the different type of reaction, chemical reaction which are taking place inside our cell. So, enzymes are considered as a metabolic regulator or biocatalyst of our cell. Then discovery. In 1878, William Cohen coined the term enzyme. William Cohen coined the term enzyme. Then another scientist, Edward Buckner, in 1897 discovered the zymase enzyme from the yeast extract. I have said in the first line, those enzyme or substance produced in the yeast, that is called as enzyme. Special enzyme name is zymase enzyme. Zymase enzyme discovered by Edward Buckner from the yeast extract. So, then another scientist, Sumner, in 1926 purified and crystallized the enzyme urease from jack bean and explained the proteinous nature of enzyme. Sumner purified and crystallized the enzyme urease from jack bean and explained that these enzymes are proteinous nature, means enzymes are made of protein. According to him, enzymes are made of protein. Protein may be polypeptide chain. Enzyme may be made of single polypeptide chain or more than one polypeptide chain. So, but here then comes to types of enzyme. Types of enzyme. We know most of the enzymes are proteins, but all enzymes are not protein. But all proteins are not enzymes. You remember, most of the enzymes are protein. All proteins are not enzyme. All enzymes are not protein. Why? Because some enzymes like ribozyme, RNA function as a enzyme, that is ribozyme. Some antibody like Abzyme, that function as a enzyme. So, all enzyme never protein. Most of the enzymes are protein, but all proteins are not enzyme. Then types of enzyme. Here exoenzyme. First one is exoenzyme. Exoenzyme means what? Those enzymes which function outside of the cell. Those enzymes which function outside of the cell, that is called as exoenzyme. Similarly, zymase enzyme, lysozyme enzyme, digestive enzyme. Here these are the enzymes, they are considered as exoenzyme. Suppose in our alimentary canal, digestive enzyme secreted from the different gland and secreted into the lumen of alimentary canal. In the lumen of alimentary canal, these enzymes digest the food material. So, my point is exoenzyme means those enzymes function outside of the cell. They are called as exoenzyme. Then comes to endoenzyme. Endoenzyme means those enzymes which function inside the cell. That is called as endoenzyme. One is the enzyme for glycolysis. Different enzymes involved in the glycolytic pathway like hexokinase, phosphohexoisomerase, aldolase. 10 enzymes are involved in the glycolysis. They are the endoenzyme because they function inside the cell. So, they are called as endoenzyme. Then another enzyme is zymogen. Zymogen is inactive enzyme. Proenzyme. Before catalyzing their reaction, they must be activated. So, zymogen is an enzyme which is an inactive enzyme, inactive precursor, proenzyme. They cannot digest the food material directly. Before digestion of food material, before catalyzing their reaction, they must be activated. Next, fourth type is constitutive gene or housekeeping enzyme. Constitutive enzyme or housekeeping enzyme. Those enzymes which always synthesize in the cell. Their requirement is always for the cell. These enzymes are called as housekeeping enzyme or constitutive enzyme, like glycolytic enzyme. Glycolytic enzyme are always required for the cell, so they are housekeeping gene or constitutive gene. Then inducible enzyme. Inducible enzyme is fifth type is inducible enzyme. Those enzymes are not always required, but when they are needed, they can be synthesized in the cell. Such type of enzyme called as inducible enzyme, like nitric oxide synthetase, nitric oxide synthetase, aldehyde dehydrogenase, they are the inducible enzyme. Means they can synthesize when they are needed. So such type of enzyme called as inducible enzyme. Then another type is isoenzyme. Isoenzyme means those enzymes are different from of the same enzyme that catalyze the same chemical reaction but chemically different, like aspartate kinase enzyme. They have different form. Then another ribozyme or RNA enzyme. I have already said all enzymes are not protein. Due to this enzyme, all enzymes are not protein. Most of the enzymes are protein. Here ribozyme, RNA enzyme, here RNA function as a enzyme. Ribonuclease P, peptidyl transferase, they are the RNA enzyme. They are made from RNA. So all enzymes never protein. Most of the enzymes are proteinous nature. Then another enzyme, ABzyme, antibodies act as enzyme. Some antibody function as enzyme. Then enzyme classification or nomenclature. International Union of Biochemistry, IUB, in 1961 classified all the enzymes into 6 major classes. In 1961, International Union of Biochemistry classified all the enzymes into 6 major classes according to their type of reaction, according to their catalyzing the reaction. But each main class again subdivided into subclass as well as subclass. But each enzyme is identified by unique digit classification number. For example, each enzyme has 2 names. One is recommended name, another is a systematic name. Suppose, for example, hexokinase enzyme. Hexokinase enzyme is an enzyme which is a recommended name. But glucose phosphotransferase enzyme whose name is in systematic manner, systematic name. This is recommendation name, this is systematic name. But actually hexokinase enzyme means this enzyme help in when glucose molecule react with ATP that result in the formation of glucose 6-phosphate. Here hexokinase enzyme is responsible for conversion of glucose to glucose 6-phosphate. But this is called as hexokinase, recommendation name, and systematic name is glucose phosphotransferase. Here hexokinase transfer the phosphate group from one compound to another compound. So systematic name is glucose phosphotransferase enzyme. But here the systematic name of hexokinase enzyme can be identified in a classification number, that is EC2711. E means enzyme, C means commission, enzyme commission. 2 for major class, 2 is the major class, 7 is the subclass, 1 is the subclass, then 1 is the serial number of the sub and subclass. Remember, E means enzyme commission, EC means enzyme commission, 2 means major class, 7 means subclass, 1 means subclass, 1 is the serial number of sub and subclass. This is the identification of classification number for a enzyme given by International Union of Biochemistry in 1961. Then comes to all enzymes are classified into 6 major classes. You can remember, I will give you a formula, you can remember better, that is 2 wheel. You remember 2 wheel. You can remember 6 major classes of enzyme, that is 2 wheel. 2 means transferase, T means transferase, O means oxidoreductase, H means hydrolysis, I means isomerase, L means lysase, and L means ligase. 2 wheel transferase, oxidoreductase, hydrolase, isomerase, lyase, ligase. These are the 6 major classes of the enzyme. According to the International Union of Biochemistry, all enzymes are classified into 6 major classes, that is transferase, oxidoreductase, hydrolase, isomerase, lyase and ligase. 2 wheel. But here major class is class 1 is oxidoreductase. Those enzymes help in oxidation and reduction reaction, that is called as a oxidoreductase enzyme. Oxidation means what? Oxidation means loss of electron, loss of hydrogen, that is called as oxidation. Reduction means addition of electron, addition of hydrogen, that is called as reduction. Here different type of oxidoreductase enzyme, that is oxidase, reductase, dehydrogenase, D means remove, removal of hydrogen atom. Here alcohol plus NAD give rise to aldehyde plus NADH2. Here hydrogen removed from alcohol group and added with the aldehyde, NAD plus reduced into NADH2. Here hydrogen atom removed from alcohol group added with the NAD reduced into NADH2, but alcohol converted into a product, that is aldehyde. Here alcohol dehydrogenase is a special type of oxidoreductase enzyme. Similarly, transferase enzyme, second type is transferase. Those enzymes transfer group from one compound to another compound, that is called a transferase enzyme. Here glucose plus ATP give rise to glucose 6-phosphate plus ADP in presence of enzyme glucokinase or hexokinase. Here hexokinase is a special type of transferase enzyme, that is transminase, transketolase, transaldolase. They are the transferase enzyme. Then another enzyme is hydrolases. Hydrolases are the enzyme that perform the hydrolysis reaction. Those enzyme catalyzes the hydrolysis reaction, that is called as hydrolases. Hydrolysis means hydro means water, lysis means breakdown, addition of water molecule with a compound and break down the compound to a product, that is called as hydrolase enzyme. Hydrolysis reaction, here sucrose give rise to glucose plus fructose in presence of enzyme sucrase, that is in presence of water, that is called as sucrase is one type of hydrolase enzyme. Similarly, lyase. Lyase means those enzyme which elimination to form double bond without hydrolysis. Those enzyme which help in transfer or elimination of group to form double bond without hydrolysis, that is called as lyase enzyme, like aldolase, decarboxylase, fumarate, citrate synthetase. Here histidine undergoes decarboxylation reaction, means removal of carbon dioxide in presence of enzyme histidine decarboxylase, that results in the formation of histidine. So histidine decarboxylase is one type of lyase enzyme without hydrolysis reaction, without addition of water molecule that converts, that release carbon dioxide, that remove carbon dioxide. That type of enzyme is called as lyase enzyme. Second is isomerase. Isomerase means those enzyme which undergoes isomerization reaction, that carry out the isomerization reaction, that is called as isomerase enzymes. Here glucose 6-phosphate is converted into fructose 6-phosphate in presence of enzyme phosphohexoisomerase or isomerase enzyme. Isomer means those enzyme perform the isomerization reaction, means one form of isomer to converts into another form of isomer compound. Isomer means those compounds having similar molecular formula but different structure, molecular structure. These compounds are called as isomer compound. Here glucose 6-phosphate is a isomer compound, fructose 6-phosphate is also another isomer compound. Here glucose 6-phosphate converts into fructose 6-phosphate in presence of enzyme isomerase, that is phosphohexoisomerase enzyme. Phosphohexoisomerase enzyme is one type of isomerase enzyme, that is isomerase enzyme, mutase enzyme, epimerase enzyme, they are all included under isomerase enzyme. Another sixth type of enzyme, that is sixth class, is ligase or synthetase. This enzyme join two compounds to form a new another compound, that is called as synthetase enzyme or ligase enzyme with ATP hydrolysis. Here pyruvic acid or pyruvate added with carbon dioxide and ATP in presence of enzyme pyruvate carboxylase enzyme, that form oxaloacetic acid, ADP and inorganic phosphate. Here pyruvic acid added with carbon dioxide means carboxylation reaction. When carbon dioxide added with a compound, that is called as carboxylation reaction. So the enzyme name is carboxylase enzyme. And ATP also added with the pyruvic acid or pyruvate. So product is oxaloacetic acid or oxaloacetate. Here pyruvic acid is a 3-carbon compound, carbon dioxide is a 1-carbon compound, that results in the formation of 4-carbon compound, that is oxaloacetate in presence of enzyme pyruvate carboxylase. So pyruvate carboxylase is one type of ligase enzyme or synthetase enzyme. Here ATP hydrolysis takes place and combines two compounds to form a new another compound. So this is synthetase enzyme or ligase enzyme. Here pyruvate carboxylase is one type of synthetase or ligase enzyme. This is all about your classification of enzyme, means 6 classes of enzyme, that is 2 hill. Okay, this is your last part of our class. Thanks for watching this video.